Good evening, everyone. My name is Amar Singh. First, I would like to thank the organizers and the session chairs for having me here to talk about my research and to present our current work. I work at the University of Kansas. Our lab is focused on developing computational methods and resources for uh, structural modeling of protein-protein interaction. Today, I will be talking about the protein-protein docking and its role in differentiating interacting and non-interacting proteins. So, as we all know that many proteins perform their functions by interacting to other proteins, and they are very specific in choice of their interacting partners. So, in the context of a cellular crowded environment, one of the most important challenges is to elucidate how does a protein distinguish its dedicated partner from the non-interactor. So, uh, there are in cellular environment there are stable interactions or transient interactions. So in all these protein complexes, is there any structural preference towards the interacting partner? That's what we want to investigate. And computationally, without any prior knowledge of the interface or interaction, can we predict the interacting partner using only the structural information of these proteins? So to investigate whether is there any structural similarity between these interactions, what we do, we extract the heterodimers from the protein data bank, and then we do the pairwise comparison. So if you have two complexes, complex A, B, and A prime, B prime, and in these two complexes, if chain A and A prime are similar and they interact two different proteins, what is the structural similarity between these two proteins? So we systematically explore the self-complementarity of the interacting proteins using the binary heterocomplexes from protein data bank. So we have about 7,000 complexes in total, and we do the pairwise comparisons. For structural similarity of the individual chain, we evaluate using the TML line, and the interface of these interactions we evaluate using MML line. Now, one protein can bind anywhere on the receptor surface. So how do we measure? Okay, they bind on the same binding site. So to measure that, the binding mode or interaction of the binding site, we measure the interaction RMSD. So interaction RMSD is basically when it's a pure geometrical characteristics. We have two pair of the complexes. First, we align these two chains and then measure the RMSD between center of mass plus minus 5 angstrom of the other chain. And then we align the other two parts and then calculate the RMSD between center of mass plus minus 5 points. So this is how we can geometrically we can measure that they interact in the same mode or not. So we explore all the available binary heterocomplexes from protein data bank. We do all pairwise comparison, and we wanted to know is there any structural similarity between the interacting partners. So we don't go any interface residue or interface information, just structural information, whether they are means radius of gyration, number of residues, or solvent accessible surface area. So in if these two, oh, sorry, these two proteins share the similarity in terms of other parameters like radius of gyration or solvent accessible area. What is the similarity between the other two? So here, one protein interacts with many other proteins. If they bind anywhere on the receptor surface, they can be of any size. So here is the on x-axis the ratio of these parameters. But if they bind on the similar binding sites, they have interaction RMS less than 5 angstrom. It means many of all of them are structurally similar in terms of length, radius of gyration, solvent accessible surface area. In terms of structural similarity, we calculate the interaction RMSD. So interaction RMSD less than 10 angstrom, it means they have the similar binding mode. So two different complex pairs, the other proteins have the similar binding mode. So interaction RMSD less than 10 angstrom. This TM score is minimum of these two TM scores in the complex pair. So we see that if they bind on the similar binding mode or binding on the similar binding sites, they are structurally similar. So what we conclude here, here is one of the example that we have three different ligands bind to a similar receptor proteins. They are structurally similar but different sequentially. They have less than 30% sequence similarity. So we conclude here, if the two proteins have highly similar folds and are known to interact via the same binding patch, it is highly likely that they bind to structurally similar protein. So it means proteins recognize each other and reveal the existence of structural preference or set complementarity towards the native partner. So computationally, can we predict or can we differentiate the interacting and non-interacting protein? To investigate that, we do the protein-protein docking. We pick different receptor proteins and dock them to a native interacting partner and cross-docking with different other proteins. Those are not interacting with that receptor. And for docking, we use our in-house gram docking software, docking algorithm. And then once we have done the docking with the docking poses, we do the structural clustering of those docking poses. So the basic assumption behind the clustering approach is that the free energy landscape of interacting partners or the native structures, they have a very deeper wall rather than the non-interacting proteins. So we do the structural clustering based on the C alpha RMSD of those docking poses, and we generate a all versus all C alpha RMSD matrix of only top 2000 predictions for each docking run. And then here we have done the highly connected subgraph clustering. You see here, first we break those based on the distance of it is RMSD here, and then we break continuing like this to make them highly connected subgraphs. So this is the clustering docking predictions. On the left side, we have average number of minima or number of clusters for average whole set of docking. The red means the interacting protein when the receptor protein docked with its native partner. And we did docking for three different other proteins which are similar in the size of the native partner, bigger size, smaller size. And we see that the clustering pattern is different for if the docked part protein is different in the size. So if a particular clustering radii, 5 angstrom say, we have the distribution of the density plot of the largest cluster is different for different size of ligand protein. So here is the one of the example with the same receptor protein in green color. It docked with the native partner and docked with three other different proteins. Those are not interacting or non-native proteins. So we see here that if it is docked with the native partner, the docking predictions are clustered near around the binding site. If it is non-interacting, they are distributed all around the receptor surface. So the conclusion is that the number of minima in energy landscape are distinguishable when the receptor protein are docked with the different size proteins, smaller, bigger compared to the interacting partner. And the low energy poses of the interacting partner cluster in a narrow vicinity of the native structure, whereas for non-interacting protein, they are all over spread the receptor surface. So if we see here, we still have some similarity. We dock with the same receptor, same size receptor protein. We are trying to differentiate these docking energy landscape as well using more sophisticated approach like atom-atom contact potentials that can differentiate these energy landscape for similar size protein as well. So the summary is that exhaustive sampling of the intramolecular energy landscape can generate more adequate set of protein-protein complexes for knowledge-based or homology modeling, where we want to have a set of proteins for templates, and we want to exclude the non-interacting proteins. So this simple and robust clustering method can generate such interacting or non-interacting data sets. I would like to acknowledge my lab head, Professor Ilya Waksa, the Deputy Lab Head, Petrus Kundrotas, and the other lab members, the KU Center for Research Computing, providing the research facilities, and the funding agencies. Thank you. I would love to have any questions. I know everyone is in a hurry. I just try to finish as quick as can.
Amar actually did a great job to get us back on schedule, so we actually have a luxury of few questions. If you have, feel free. Don't even need to run to microphone, walk over. We have 5 minutes within the schedule. While waiting on questions, I have at least two questions.
Yes, sure. Go ahead.
So all examples you showed, they demonstrate analysis of two domains interaction, like one domain, one partner, another second partner. How do you deal with multi-domain proteins? Because your computation scheme seems to be relying on the center of mass or something. So are you splitting it in the separate domains and then analyze?
Yes. So the similar binding mode we calculate through the interaction RMSD, and it is pure geometric. So if you have different binding mode, one can rotate, and then it will have the similar binding site, but interaction RMSD will increase. So that's your question. So how to deal with that? So what we do in that case, instead of using the interaction RMSD, we calculate the receptor structural similarity and the interface similarity of that receptor from the binding partner. So suppose if we have this receptor protein, one binds here, other binds here. In that case, we calculate the structural similarity of this receptor protein and the structural similarity of the interface. So in that case, the domain of the interacting partner doesn't come into picture. So that well, I don't have the result for that one, but you see these are here. So because of the orientation of the ligand protein, the interaction RMSD will high, but still they have structural similarity higher than 0.5, the TM score.
Oh, we have from the.
Can you just go forward and try to catch a reference where you talk about the shallow wells or deep wells?
Oh, energy landscape.
Yeah.
So it's from Ilya Voxer. So
Yeah. Thank you.
it just the picture from the same reference. It is when this picture shows for native, non-native in terms of refinement. When you have true native in same binding site, we call it native, and non-native. So native predictions are always confined in a well-established well. So that's the basic assumption. Here I am considering native, non-native in terms of interacting, non-interacting. So.
All right, so questions from virtual audience. MM align does not align only the interface. It is a full-length complex structure alignment. Would an interface structure alignment program such as PC align or I align be more appropriate for IRMSD calculations? So essentially,
yeah, here we see that if we align the individual chains, TM align works best. But if you have two chains in the structure, align we use to do that one. So here I am using for the structural similarity between the interface, I use the align. So that was the question that align cannot be used for individual chain. Yes, I use align for the interface where both structures are considered for the interface.
Well, the question was that it actually uses the entire chain. So you're still able to localize the alignment for two interface?
Sorry, I did not get that one.
So MM align does not align only the interface. It is a full-length complex structure alignment.
Yes.
So they suggest you to use different aligners just for.
Oh, I extract the interface, and then I use that structure only. Maybe
even though it's a discontinuous then, right?
Yes. So I did not align the whole structure. I first extract the interface and then do the align. If I have the whole structure, yeah, it's true. It will align the whole structure, not only the interface.
Okay. So second question: when you do RAM docking, do you use the upper structure or the bounded structure?
Bound structure.
Bounded.
Bound conformation. For the docking of the interacting or non-interacting, I use the bound conformation. That was the question.